All right, welcome back to this tactical intellectual channel. The topics for this presentation: Dr. Selina Brace, Cheddar Man, and last but not least, the HIrisplex or HIrisplex tool. They use that in forensics. Now, the content that I am drawing from comes from this video right here. I will leave a link to this video in my description so you can take in the full interview. It's very thorough, as well as being intriguing and insightful, to use one word if I have to sum it all up. To start off my presentation, I would like to play a clip from the Evolution Soup interview with Dr. Selina Brace. You can refer to this as a mini Scholars Corner segment only because I delve into her background, her field of study, what she's all about, etc. So I hope you guys enjoy this part, and here we go.
Dr. Selina Brace, thanks so much for coming on to Evolution Soup from your home in Hampton, UK. You are a researcher in the Earth Sciences Department and manage the London Natural History Museum's Ancient DNA Laboratory, working with ancient and degraded DNA in order to investigate evolutionary processes. You are one of the team that worked on the famed Cheddar Man skeleton, which is what we are going to be discussing today.
So that's a little bit about her. Of course, she was part of the team of scientists who did some work on Cheddar Man. Now, you may not be familiar with who Cheddar Man is, so let me play this clip right here discussing who Cheddar Man is, and of course, Dr. Selina Brace with the commentary. And I find that fair and appropriate. So let's go ahead and hear what she has to say.
Okay, time to talk all things Cheddar Man. Now, for anyone who is unfamiliar with this story, Cheddar Man is the oldest complete skeleton ever found in Britain, discovered in Cheddar, Somerset, hence the name. So first of all, what can you tell us?
What we do know about him is that he died in his early 20s. We of course know that he wasn't 40 or 80,000 years old. He was, in fact, he died 10,000 years ago. He has been radiocarbon dated many times over now, so we are very clear on the date when he died, 10,000 years ago. This means that Cheddar Man was alive during the Mesolithic period. So the Mesolithic period is a time before farming. So this is the time of the hunter-gatherer. So this is when people were living in these portable animal skin tents, or were taking shelter in caves, perhaps like Cheddar Gorge, but they certainly didn't stay in any one place. So they were a nomadic people. They would have been living off the land, moving with the seasons. They likely hunted animals such as auroch, deer, wild boar. We know the kind of tools that they used. We find axes and microliths and spears from this period. Britain itself would have been more like a woodland at this time. The landscape would have been more forested woodland. And interestingly, Britain was still connected to continental Europe at this time. So some land masses are connected by land bridges. And so this is basically during colder periods, during glacial periods, water from the oceans is taken up into glaciers, so sea levels actually drop, and they drop so considerably in certain places that actually land is exposed, and you have a land bridge between two different countries, which is what happens to Britain and Europe during this time. And that land that's exposed, that's in between, is called Doggerland, and that remained until in the region of 8,000 years ago, when it was once again flooded. So I guess we could say that the Britain that Cheddar Man lived in would have looked quite different to how we know Britain today.
So was he the only skeleton in the cave, or has anything else of interest been found?
So no, Mark, he's not the only skeleton in the cave. And though he certainly is the oldest, most complete skeleton, he wasn't the first Englishman either, because fairly close to where Cheddar Man was found, there's another group of, we call it an assemblage of fragmented skeletal remains. The number of individuals here is not certain because these bones are highly fragmented. They're not complete like Cheddar Man. It's lots of small fragments of human remains. But based on bones that are identified, the developmental age of those bones, the size of the bones, and the sex of those bones, we think that the minimum number of individuals that this assemblage represents is 6. And this would comprise of 1 child, 2 adolescents, and 3 adults. These bones have been radiocarbon dated to much earlier than Cheddar Man. So these bones are around 15,000 years old. So this is from the Paleolithic period. Mm-hmm. And these people would have had an even harsher existence. The climate would have been colder. Indeed, it got colder still in the UK, and the UK was in fact uninhabitable by people between the times of the Paleolithic people and then 10,000 years ago when we see Cheddar Man.
I really appreciated what she said, the background information that she gave on Cheddar Man, his age, the point in time in which he lived in. Then was it Mesolithic? So she also gave interesting background on the geography of Britain or the UK. Then it wasn't a collection of islands, but it was still connected to continental Europe. That may definitely be a hard concept for us in our modern times to conceive, but hey, it is what it is. So that brings me to my next segment or clip that I want to showcase to you guys. I thought about skipping this one because it's all about the HIrisplex and Cheddar Man, but some people may be wary of her method of extracting DNA or what she really does. Some people may question her credentials, so let her explain her field of expertise. Hope you guys enjoy it.
Well, in 2018, DNA was extracted from the skeleton of Cheddar Man and revealed some pretty interesting results. But before we get into those findings, talk a little bit about ancient DNA and the techniques that are now used to examine genomes and uncover details about individuals like Cheddar Man.
So I am an ancient DNA researcher. So my job is literally to get DNA out of dead things. But to clarify, ancient DNA doesn't really mean or doesn't have to be about age. It doesn't have to be about the age of the DNA because as soon as an organism dies, the DNA in that organism will begin to break down. And so depending on the conditions that organism is left in, depends on how fast that DNA will break down. So generally speaking, if a sample is in a cold place, the degradation process slows and it takes longer for the DNA to break down. And if it's in a warm environment, then that process speeds up and the bonds holding the DNA together break down faster and the DNA becomes fragmented and shorter quicker. I work with a variety of different types of material, but it's typically bones or teeth, skin, sometimes hair, or even coprolites, so like animal dung. I work in a specialized ancient DNA laboratory at the NHM. But in terms of the sampling, once we get the material, which quite often comes from museums or from archaeological, zooarchaeological sites, I take it into the lab. If it's something like skin, then I would cut it into very small pieces using a razor. If it's bone, then I drill a small, like a 3 millimeter hole into the bone using like a dentist drill. We use a dentist drill because that goes at very, very slow speeds and we don't want the drill to heat up and destroy the DNA. So we drill into the bone and release a small amount of bone powder. And then it's this bone powder or tissue that then I use to extract the DNA from. I do this initially by breaking down everything that isn't DNA with enzymes. And then we basically bind the DNA to what's like a piece of silica. And then we wash out everything that's not DNA, again, with chemical solutions. And we wash it all through so that we're left with DNA. After that, we essentially prepare the DNA for sequencing. So again, this is some chemical reactions, but then we put it on a sequencing machine that will sequence the different fragments for us. So then we basically get lists of A, C, Gs, and Ts, and we use bioinformatics to put all those fragments back together again to create a whole very long string of A's, G's, and T's.
And what was the actual bone for Cheddar Man that you would have had to drill into? Is there a specific part of him?
Yes, there is actually. So with Cheddar Man, I actually drilled a very small hole into his petrous. His petrous is the inner ear bone. And that is actually it is the densest bone in the body. And so it actually seems to, it doesn't necessarily preserve the DNA, but it's more that it protects the DNA from outside influences. So you tend to get longer pieces of DNA and more endogenous DNA. So more of the DNA you're interested in, more of the human DNA in a petrous bone than of like soil bacteria, which is what we get an awful lot of when we're doing this type of work. So yes, so in this case, it was the inner ear bone, the petrous bone.
Now, let's go into the main event. Let's go into the portion of my presentation that we are all here for. The HIrisplex, or as I like to refer to it as, the HIrisplex tool. And let's let her explain the importance of it, because I know the detractors and denigrators and just regular everyday people, they probably never even heard of the HIrisplex or HIrisplex tool. So without further ado, let's let it rock.
Yeah. So what were the results of the DNA sequencing? What did Cheddar Man look like, and how can we be sure of these findings?
So in the case of Cheddar Man, we were actually sequencing his DNA as part of a much wider study that was looking at many different individuals to explore the transition in the UK from Mesolithic hunter-gatherer to that of the Neolithic farmer. And the results in the broadest context from Cheddar Man helped to show that the genetic signature of a hunter-gatherer was very, very different to the genetic signature of a Neolithic farmer, meaning that different groups of people had moved into Britain at the same time as farming practices. So a movement of people, not a movement of ideas, is the broad context of what we were looking at with Cheddar Man results. But in relation to Cheddar Man as an individual, we showed several different things. One is that he did not have the genetic variants that would have allowed him as an adult to digest milk. So he would have been lactose intolerant. This in itself is not particularly surprising because obviously this was a time before farming and before domesticated animals.
Right.
So he would likely not have been exposed to any dairy products anyway.
Key point: lactose intolerance. We know what type of people on this planet are mostly lactose intolerant in large numbers. But if you're still not convinced, just stay tuned.
In relation to Cheddar Man's phenotype, or what he physically looked like, we described Cheddar Man as having dark brown to black hair color, blue or blue-green eyes, and dark or dark to black skin pigmentation. So I'm just going to focus straight in on the skin pigmentation result because this is what's caused the most issues for people. So human skin pigmentation is a very, very complex trait. It's what we call polygenetic, meaning that there are many genes and many gene variants that affect it. We probably don't know all of them, and we certainly don't know how they all interact. Indeed, it's fair to say that in recent studies from modern African populations, they found more than 100 places in the human genome where people vary, where there are variations that can be associated with skin pigmentation. So to sort of clarify, these are sites in the genome where I might have a C and you, Mark, might have a T. And when individuals have a different base at a single site, we call it a single nucleotide polymorphism, or SNP for short.
All right, you guys already know that from my migration video presentations. I have 5 of them, so I think it's the third one. He goes in depth in SNPs or SNPs, and her point regarding Africa and the genetic variants, I think Africa, that continent, has so much genetic diversity, more than Europe, well, modern day Europe, more than modern day Asia, and so on. So let's go ahead and continue with the video.
You quite often hear people talking about SNPs, and that's what they're talking about. So there are a lot of these SNPs associated with skin pigmentation. Some of these variations are found specifically within genes where we know the effect of those genes. And the effect of those genes are, they can be on genes that make either a different type or a different amount of melanin. Now, melanin is the body's natural skin pigment. And so this will affect the amount of skin pigment that an individual produces.
We all have it.
Yes, we all have those genes.
Now, regarding melanin, you have pheomelanin and eumelanin. Eumelanin is what the Negro people have, and pheomelanin is more reddish. You know, that's what everybody else has in large numbers.
And these genes have names, and the important ones are ones like MC1R. And in Europe, specifically in Europe, there's a suite called the SLC genes, 24A5 and 45A2 are the key ones, and another one called TYR. But what's interesting is, so whilst there are many, many SNPs associated with skin pigmentation, the most recent studies are indicating that only a few of these SNPs, only a few of these variations seem to be responsible for the largest effects in skin pigmentation, so the largest differences, and that these SNPs are shown as signals of selection in populations through time. So with this kind of background in mind, we can go back to Cheddar Man and explain exactly what we did there. So what we did is to apply a specific tool called the HIrisplex S system. Catchy name. But it's actually used to predict the eye, hair, and skin pigmentation. It's a tool that's designed to be used for forensic cases and to predict pigmentation based on variations, so based on SNPs in an individual's genome. So the eye color test is based on 6 SNPs, the hair color is based on 22, and skin pigmentation is based on 36 different SNPs. So this is not the 100 SNPs that you might find in a modern African population, but 36 SNPs that are known to have a large effect in Europe. So these are these ones like these SLC 24A and 45A2 that I was talking about.
Is this starting to become clearer to you guys now? A lot of people don't just make this stuff up. You know, this is not a quote unquote woke narrative, but we want to come towards the truth as best as we can and uncover the lies and bad narrative.
And what this tool actually does is make a prediction based on a statistical model that has been trained on empirical, so real data. And what it does is it gives a probability score that reflects the uncertainty, because of course this is actually very difficult to predict. So it reflects uncertainty by giving a probability score, and these scores are then used to look at very broad categories of pigmentations, specifically a thing called the Fitzpatrick scale, which is used in dermatological skin assessments, and it categorizes skin pigmentation into 6 broad categories that range from very pale white right through to very dark brown black. And it was this tool that we used to generate probability scores to predict the results. So when you say, can I be sure? Can I be 100% sure? No, because it's a very complicated trait, and it's always possible that a genetic variant existed in the past. So there was perhaps a SNP in the past that we don't see any longer today that would have conferred a different skin pigmentation. However, as there seems to be a very strong selection for lighter skin pigmentation in more northerly latitudes, I think we're going to talk a bit more about this in a moment, I think, then one would imagine that if this variant had existed in the past, then this too would have had a very strong selection for it to continue in Europe, in northerly latitudes. And so we would still see it in modern populations. So in the absence of these unknown genes, but based on the genetic data that we obtained, the genetic variants within genes that we sequenced, the probability scores from these modern day forensic tests that we applied, personally, yes, I remain confident in our assessment that Cheddar Man had dark or dark to black skin pigmentation.
Very good point. For everybody who want to detract and say that one group of people, they're how would you say, they have a inferiority complex because they try to hop on. We all know what I'm getting at here. The narrative is that, okay, this group of people, they had, due to their deep-seated inferiority complex, they want to claim other and hijack other people's cultures, when really it's the other way around. And we're just uncovering everything here. But she had a great point about that probability score. She's not 100% sure, because you can't be 100% sure. But if there was some unknown SNP that had a different tone of skin color, you will probably more than likely see it in the modern population. But you don't, so come on, probabilities here. Let's go.
And I would also finally add that this wasn't actually unusual. So there are other Mesolithic hunter-gatherer individuals whose genomes have also been sequenced from across Europe, and they too have been identified as having this darker skin pigmentation coupled with this very light eye combination.
Bingo, all across Europe. Should I play this again? Let me go ahead and play it again. Let me play it again.
Also, finally, add that this wasn't actually unusual. So there are other Mesolithic hunter-gatherer individuals whose genomes have also been sequenced from across Europe, and they too have been identified as having this darker skin pigmentation coupled with this very light eye combination. So to my mind, no, we really shouldn't even be that surprised by this result.
All right, so I had to pause it right there. Let that marinate, digest that information. Now, going forward, the host of this stream here, he's going to employ devil's advocate regarding that skin pigmentation. And we all know the, how can I put it? It's kind of conjecture field, but the point about vitamin D deficiency, he's gonna bring that up. So I'm gonna just play it right here.
I'm going to play devil's advocate here. Wouldn't an individual with skin as dark as that have had suffered health problems in those cold northern latitudes, especially with vitamin D being so scarce?
Well, Mark, yeah, this is actually really, really interesting because, of course, skin pigmentation is known to have a very high correlation with geographic and environmental variation. So populations at lower latitudes, those close to the equator, have a darker skin pigmentation, and those at higher latitudes have a lighter skin pigmentation, sort of traditionally. So scientists believe that this is likely an adaptation to ultraviolet radiation from the sun. So the closer to the equator you live, the more UV light you'll receive. And so with high levels of UV light, your skin needs to be protected from the damaging effects of UV, such as burning of the skin and skin cancers. And a darker skin pigmentation, which, as we said before, I think is caused by a greater concentration of melanin, that body's natural skin pigmentation. Melanin itself absorbs that UV light and is thought to protect skin cells from UV radiation damage. However, as you move further north, then the darker skin pigmentation becomes less important in terms of protecting the skin from UV light, as the amount of UV light is obviously decreasing. It gets a lot colder and a lot darker. So instead, it's thought that the increased levels of melanin actually have the converse effect, and they can have a detrimental effect on human health, because whilst melanin actually absorbs UV light, this reduces the amount of UV that's available for vitamin D synthesis. So vitamin D is very important. It's important for musculature, skeletal health. Deficiencies cause abnormalities in calcium, phosphorus, and bone metabolism. And this can lead to problems with bone and muscle weakness. In children, it leads to skeletal deformities, also known as rickets. And vitamin D is rather unique because it can be made in the skin from exposure to sunlight. So the increased levels of melanin in darker skin pigmented individuals reduce the skin's ability to produce vitamin D from sunlight. So as you were saying, a question that we often get is how would Cheddar Man survive in Britain with darker skin pigmentation when this would have reduced his ability to produce vitamin D? So there are several sort of answers to this. One is that Cheddar Man would have had a very different lifestyle. He would have had a much greater exposure to sunlight. He would have been outside, one would imagine, for most of the daylight hours. He would possibly have had less covering and therefore would have had more skin even exposed to the sunlight. The second point is that whilst vitamin D is synthesized through sunlight, it is also obtained through the diet. Sources of good or high levels of vitamin D can be found in oily fish, such as salmon, red meat, liver, and also mushrooms. Cheddar Man, we know, or we think, had actually a very good diet because his teeth are very healthy. And as a hunter-gatherer, his diet would have likely included such foods as would have been high in vitamin D. Now, there's one other point here that's possible, and this is some work that came out recently by Hanel and Karlberg. And this suggests that there are variants in the genome, so this is back to these SNPs again, that can be associated with improved vitamin D transport and metabolism. So these might be variants that actually boost vitamin D efficiency in different individuals. So this would mean that individuals at a higher latitude could kind of negate the problem of low vitamin D intake not through lighter the skin and improved vitamin D from sunlight, but instead through improved ability to process vitamin D and its metabolites. I should stress this is not my particular area of expertise, but it certainly sounds really interesting to me, and it could be worth exploring further as another alternative explanation as to how people like Cheddar Man would have survived in these northern latitudes in light of vitamin D.
And those alternative explanations, that's something that needs to be brought up instead of just relying on the okay, he's dark skinned, he's living in northerly latitudes, he's gonna be deficient in vitamin D, and saying that the probability of that is, low. That's bad science. That's kind of bad faith, really. And also a point about melanin that she had mentioned. I have this book right here. I'm gonna put up a picture of the cover here. Melanin, the Master Molecule by Arturo Solis Herrera. He's a Mexican scientist. If he's not, then I'll put the correct information on there in a post-production edit or a slide. But melanin does far more than just absorb light. It absorbs other frequencies too. So, and convert them into other things that's good for the body in general. So I'll probably show some information from this book here to support my claim. But in the meantime, let's go ahead and move forward.
Well, some people watching this are now probably wondering just how it was that lighter skin tone evolved in those northern latitudes. So how did lighter skin come about?
Hopefully I've explained what we know and don't know about the genetic basis for skin pigmentation, and also something about the potential selective pressures or advantages that could have led to a lighter skin pigmentation in the more northerly latitudes. But to address how we think that these gene variants spread in a more physical sense is probably best explained by the idea and the facts of these physical migrations of people. Now, the first migrations of people carrying these lighter alleles come from the Anatolian farmers. So these are the people from the Near East, so from modern day Turkey, who first started farming and who spread across Europe starting around 8,000 years ago. The genes or the gene variants of these early Neolithic farmers indicate that they would have carried these light or intermediate skin pigmentations, and they also had dark hair and also dark eyes. This migration was then followed later by a second wave of migrating people. These people originated from a region between the Caspian and the Black Sea, and these are the people known as the Yamnaya or steppe pastoralists. These people migrated also in an easterly direction across Europe, starting around 5,000 years ago, and they too carried these gene variants that we associate with lighter and intermediate skin pigmentations, and it's these people that also brought with them the variable eye colours and some of the blonde hair, and so that combination of lighter skin and blonder hair that is more common today. And so from genetic studies of both of these groups, so looking at lots of individuals, these Neolithic farmers and these Yamnayan steppe pastoralists, we can see that these lighter alleles were actually what we say were fixed in these populations, which means that the majority of people were carrying them. And remember, we're talking about Europe here. And so the selective pressure from these northerly latitudes with this reduced UV, low vitamin D, they were farmers, so they didn't have that varied diet like the hunter-gatherer that I was talking about earlier. They actually had quite a limited diet, so they probably had low vitamin D, they would have had vitamin D deficiencies. So this would have really pushed in these instances for the selection for the lighter skin pigmentations to an even higher level, making it more fixed in these populations.
All right, all of that was interesting. So we have the information here ourselves, so we can no longer just claim that the early Europeans were all just modern day white folks, what they call it, the Neolithic farmers, then the Yamnaya steppe cultured people, that had these fixed, um, I want to say, all of this stuff right here, these traits were selective, and it was, like she said, fixed within their populations. And that's a far cry from the original melanated Negro Europeans. And like you say, some people gonna look at this and be like, no, 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 put the fingers in their ear to try to blot out the sound of my voice. But hey, the facts are facts, man. The evidence is there. So it is what it is. Let's revisit this Selina Brace clip here.
And a darker skin pigmentation, which, as we said before, I think is caused by a greater concentration of melanin, that body's natural skin pigmentation. Melanin itself absorbs that UV light and is thought to protect skin cells from UV radiation damage. However,
is a pretty appropriate segue into what I want to say. Now, earlier I played that Selina Brace clip of her saying that melanin protects the body from harmful UV radiation. Now, I said it does more than that. Here's the book, Melanin, the Master Molecule, and let's skim through some factuals right here. On this page, it says the melanin molecule is very similar in all species, and also its function transform light energy into chemical energy, disassociating the water molecule as chlorophyll in plants. On this page, it mentions how the constant presence of melanin suggests a dominant role not only in life itself, but from its origin. On this page, it says the amount of melanin in the skin is determined mainly by the amount of light. In places of plenty of sunshine, the amount of melanin increases for, size or sees this light energy, but at the same time regulates the amount of light which penetrates inside the body, since up to bone and muscle need light. In dim light, such as Northern Europe, the amount of melanin in the skin is less, so that it can pass through the tissues, nourishing them. Now, on this page is the main point that I referred to when I said it does more than just absorb UV radiation. It says melanin can also absorb all wavelengths of the electromagnetic spectrum, from radio waves to gamma rays. The mechanisms of this astonishing fact is not understood. Therefore, it is said that melanin is the darkest substance known. So in reality, we're dealing with thermodynamics, and thermodynamics, if you don't know, is the transformation of energy from one form to another. Thermodynamics. I'm not getting into that now, but probably later on in a future presentation. We've reached that time where the conclusion is near. I hope you guys enjoyed it. I hope you took in some knowledge. The full video, I will link it in the description. Now, what video am I talking about? Well, the Dr. Selina Brace interview. I will link that in my description. And hopefully you guys took in a knowledge on melanin from the book Melanin, the Master Molecule. And I'm stumbling over my words. I've been editing all day. I'm done finally. So anyway, I hope you guys enjoyed the video presentation. I have to get off this laptop and breathe some fresh air, go outside and run a little bit. Anyway, guys, take care.